So Doc, please tell us about yourself. Where are you from? What and where did you study?
Okay. So I'm Dr Siyoum. Originally I'm from Eritrea. I did my BSc degree in plant science back home at the University of Asmara. And my MSc and PhD studies here in South Africa at the University of Natal and University of Pretoria, respectively. Yeah.
So Doc, what sparked your interest in your field of study, from your plant science up until your PhD?
Okay. So first, I loved nature and plants. So I did plant science for my first degree. And later, my interest in pathogen detection started when I learned about the quarantine service that was provided in one of the port cities back home where I grew up. I desired to know more along that line, and when the opportunity came for my MSc study, I chose plant pathology. Later, it expanded to microbial ecology when I joined Prof. Corsten's group for my PhD study. During my PhD, I explored the microbial dynamics of mushroom substrates, and currently for my postdoc work, my research is my microbial ecology focused research on mushroom substrates, postharvest pathology, and biocontrol.
So Doc, with your array of studies that you've done, so who would you say are specific scientists or researchers who have influenced your work?
So many have influenced my work, such as my supervisor during my MSc, Prof. Mark Land, at the University of Natal, University of KwaZulu-Natal now, and Dr. Karen Sarich, my PhD co-supervisor. However, the biggest influence comes from Professor Lisa Corsten, my PhD and postdoc supervisor. She introduced me to the fields of microbial ecology, postharvest pathology, and biocontrol research.
So Doc, can you please share a memorable moment or a breakthrough moment in your career?
Okay, so one of the memorable moments was as a group when we visited mushroom farms regularly and did pathogen surveillance, and we could detect hotspots of pathogens before disease manifested or progressed.
And what are the challenges you faced in your research, and how did you overcome these challenges?
So at times there were some microorganisms didn't grow in the lab or got contaminated, and PCRs didn't work. And these challenges were overcome by optimization of methods and troubleshooting, which is part of research.
So in acknowledging the PCR is not working or the microbes aren't growing, so how do you stay motivated and inspired in your career in general?
Okay. So the realization of creating a solution to a problem with our research always kept me motivated.
Doc, how do you balance your professional and personal life?
So I try to balance my professional and personal life. First, by committing myself strictly to work during work hours, sometimes adding a couple of extra hours. Secondly, when I am at home, I attend to my family closely with some flexibility to engage in urgent work stuff.
So Doc, would you kindly share a funny or an interesting story from your research or lab experience?
Well, not a story, but an interesting fact is mushrooms are fungi and they can get infected by other fungi, which makes disease control very difficult because the growth requirement for both is similar.
What advice would you give someone starting a career in your field?
So I would advise young people starting a career in similar field or any field for that matter, to work hard and use time efficiently. Also to spend enough and more than enough time reading and doing basic lab research. This will pay off a lot later.
So Doc, how do you think your field of study will evolve in the future, and what excites you about this evolution?
So with the current advancement in technology, I think identification and microbial and pathogen identification or detection will be far more quicker and cheaper. Apps are being used to identify some different plant diseases currently, and almost all plant diseases will be identified using apps in the near future. And our understanding in microbial ecology and microbiome characterization is increasing exponentially, and it would excite me, the application in plant disease.